My name is Shailendra Singh Sera and I am Assistant Professor at Department of Biotechnology, Faculty of Engineering and Technology, Rama, University, Kanpur. So, today my topic of lecture is the animal cell culture. In this topic, we are going to discuss about the overview of animal cell culture. What is animal cell culture and what are the basics requirement of animal cell culture and how you are going to grow it in the lab and what are their application. This lecture video will prepare you for the more advanced topic on the later lecture. Let us start with the definition of animal cell culture. So, now the question is what is animal cell culture? Animal cell culture basically refers to the cultivation of cells from the outside the body in artificial environment. Let us start with the history when this animal cell culture was first established. The history dates back to the 19th century. In first, its foundation was laid down by a scientist named Joly in 1903, which was further carried out by the scientist named Harrison and Carroll. Harrison did it in the 1907 and Carroll did it in the 1912. What Carroll basically established is that he used the frog cell, isolated the frog cell from the frog body and used different method to grow outside into the lab condition. This experiment proved that animal cell can be grown outside the animal body or outside the body in an environment condition, provided the right growth condition. And what do you understand by growth condition? Growth condition means proper set of environment, temperature, pH and nutrient media. Let us see further about the laboratory facilities that are required nowadays for growing animal in lab condition. So, basically for a small educational laboratory, we need only 5 to 6 basic equipment. First one is the CO2 incubator. As we know, animal cell requires CO2 for their growth, so we need a CO2 incubator. Second one is the inverted microscope for routine analysis of the growing cell. Media is extremely important, which provides the right set of condition, growth requirement, nutrient for growing animal cells. And laminar hood. Laminar hood is one of the important equipment. Why? Because it provides an sterile area for transfer of animal cells which you have isolated from the animal body. And the last one I want to mention is that the requirement is extremely similar to the microbiological cell growth. If anyone have basic understanding or have earlier worked into the microbial lab system or have grown the microbe in laboratory, they already know that for growing microbes we need we need media, then we need a sterile media, that is autoclave for sterilization purpose and all those things. All the techniques are the same. What is the difference then? The difference lies in the aseptic requirement. In animal cell culture, we maintain a stringent aseptic requirement, which we do not do into the microbial culture. Here we need a clean room, we need a closed room, which should be away from the mass movement. Not a single particle of dust should be there. So, stringent aseptic condition is the foremost requirement for growing animal cells into the laboratory. After the requirement, let us see laboratory facility. It can be described into 3 parts. One is the sterile area, second one the tissue culture equipment, and third one is the storage area. What do you mean sterile area? Sterile area means your atmosphere should be neat and clean, free from the contamination. You should use proper personal protective equipment and also you should use laminar flow. Tissue culture equipment, In this we describe 4 or 5 basic requirements: autoclave, inverted microscope, CO2 incubator, and refrigerator. Basically it is not used, it is used for storage. And last one is storage area. Storage area can further be categorized into storage area for the storing your lab wares, chemicals, and for cell line storage. Now the question is, what is cell line storage? Suppose you have taken out a cell from some animal source or human body, you have grown it in the culture. After that you have characterized it. Every time you are not going to do the same process because it is very time consuming, and the chemicals that are used in animal cell culture are very costly. To protect our time and money, we store the cell line. For that we need a cell line storage, and in the cell line storage we use liquid nitrogen or cryopreservation method for storing the cell line. This will be discussed in much more detail into the second lecture video. Third one is the cell culture condition. Till now we have seen about the history, laboratory facilities. Now we are going to see about the cell culture condition. What are the conditions that are required for growing animal cells into the lab condition? So for that we need serum and media, pH, and incubation. Serum and media. Basically the purpose of serum and media is to provide the right physiological environment, nutrient, growth factor, vitamins, amino acid, mineral, and trace element for growth of animal cells. Media can further be categorized into 2 types: natural media and synthetic or artificial media. Now let's discuss first natural media. What is natural media? Natural media is the one which are isolated from biological fluids which are occurring naturally. Then example are serum, calf serum, bovine serum, fetal serum. Second one is the artificial or synthetic media. These are the media which contains organic and inorganic nutrient, which we prepare it by mixing several vitamins, growth factors, and then preparing it in the lab. And the example of some artificial media can be RPMI, DMEM, and DMEM Dulbecco modified eagle medium, or basal salt solution. These are some of the example of the artificial media. Second is the pH. pH is extremely important. Why? Because if we compare it with the microbiological cell culture, different microorganism grows at various range of pH. Some grows as 1, some grows as 2, some grows as 3, and some grows at even at 10 or 12. But in animal cell culture, the pH range is very narrow. It should remain between 7.1 to 7.4. In some book, you will find 7.2 to 7.6. It means basically you have to be near the neutral range of the pH. Third is the incubation. Incubation, we are growing animal cells. So the body temperature, our body temperature is 37 degrees centigrade. So incubation temperature will always be 37 degrees centigrade, whether you are growing animal cells or human cell. One of the most important component of media is also phenol red, which is not listed here, but phenol red is one of the important component of media. Now the question arises, what is the purpose of mixing phenol red? The phenol red acts as a pH indicator. As I have already told you that the pH should be between 7.1 to 7.6, yeah, 7.2 to 7.4, around neutral. Any deviation will kill your animal cell. So you need to know if there is any deviation into the media of growing cells. So the phenol red will change its color if there is any deviation towards the alkaline side or the acidic side. You will commonly know that your animal cell culture has different media. So you can just stop it, take it off from the incubator and change the media. The change of media can also indicates about the contamination, whether it's bacterial, fungi or mycoplasma. Mycoplasma contamination cannot be detected by the change of color. Basically, fungi and bacterial contamination can easily be detected. So, phenol red has dual function to change to indicate the change of pH from neutrality, and it can also indicate the contamination, bacterial and fungal contaminations. Fourth one is the various system of cell culture. Till now we have discussed about the history, laboratory facilities, cell culture conditions. Now, what are the methods to grow them? So, basically there are 4 to 5 methods. We can grow it as a monolayer culture, we can grow it as a suspension culture, we can grow using cover slip or slide culture, we can use hanging drop culture method, and lastly we can also use flask method. What now we will give, I will give some basics introduction, and we will discuss it in the next video in detail when we will discuss about the methods of cell culture. So, monolayer culture. Basically, what do we monolayer? As the name suggests, monolayer means just a thin film. The cells will be growing just as a thin film. There will be no three-dimensional or thickness. Thickness is very less. Suspension culture, cells growing in aqueous medium. Cover slip or slide culture method. In the cover slip or slide culture, there is a one slide like that, which has some hollow. We put our plasma cloth here and cell, and we put it using cover slip. So, this is a specially designed cover slip which comes for cell culture method. Hanging drop. What does it mean by hanging drop? As the name suggests, we have a special equipment like that. We put our media here and the cells grows hanging just like a ceiling fan. If you have ever watched the ceiling fan, it is hanging from the roof. So, the cells will grow just like hanging, and the nutrient will be evaporated from here, and here they will get the nutrient. Last one, fifth one is the morphology of animal cells in culture. Morphology means appearance, shape and size. So, since we are growing outside of the body, it is bound to be differ from as it is growing in the body and growing outside the body. What are the basic types of cell morphology shown by animal cells in culture? Basically, we can categorize in 3: fibroblastic, epithelial and lymphoblastic. Fibroblastic and epithelial are adherent cell culture type. Adherent cell culture type, and fibroblastic looks like a spindle shape. Epithelial looks like polygonal shaped, and lymphoblastic is a round shaped. And lymphoblastic always grows in suspension culture. Now, there I have used one term, adherent cell. Now, you should ask, what do you mean by adherent cell? Adherent cells are the cells which can grow when they are attached to any substrate, and non-adherent cells are those cells which does not require any attachment to the substrate. And basically, adherent cells are grown as a monolayer, and non-adherent cells are grown in a suspension culture. Now, you have grown all the cells. Now, you have to continuously monitor their growth. How you are going to monitor their growth? So, basically in a normal cell there are a lot of technique, but since animal cell culture is extremely costly, so we want to utilize some non-costly technique which are easily available, and which is, on the other hand, they are highly sensitive also. So, Hema Cytometer and viability assay. Hema Cytometer first discuss. Hema Cytometer is specially designed slide where you have gridded structure. You load 10 microliter of your sample over it and see it into the microscope and count the cells and do the calculations. Viability assay. Viability assay means we have to ascertain whether the cells are live or dead. This is what viability assay means. Then how we are going to ascertain this? In the viability assay, the most commonly used is the trypan blue assay. Trypan blue is a dye which are taken only by the dead cells. It means if your cell is dead, it will appear blue into the microscope. If your cell is not dead, it will appear transparent. So, dead cell, then now the question arises, you can ask why dead cell appears blue? Dead cell appears blue because dead cell, after cell death, the permeability, the cell wall or cell membrane get disrupted, okay, and the dye enters into the cell and colors it. Whereas in living cells, the cell membrane is intact and the dye is unable to penetrate, and hence it is unable to stain it and it looks transparent. Seventh point is the characterization of cells in culture. Now, the question is what do you mean by characterization? This is one of the most important topics anyone dealing with the cell culture, because there are number of cell lines into the lab. Someone is isolated from monkey, some from human, some from this animal, some from that animal, some from that animal. How you are going to ascertain this is a monkey cell line, this is a human cell line? So, here the characterization comes handy. So, there are various techniques of characterization. Someone is a karyotypic characterization, where we see the cell numbers, chromosome number, sorry, not the cell number, chromosome number and chromosome shape and size. So, characterization basically means identification of cell lineage or identification of surface markers. Every cell has unique surface marker which cannot be found on other cells. So, just identify that unique surface marker will give us the idea that this cell is a herd cell, or this cell is coming from the human cell, or this cell is coming from the monkey. Okay. Last one is the application. There are a number of application of cell culture. I have only written 4 or 5, but there are a number of that. One of them is tissue engineering. Here tissue engineering means we are using cells, animal cells for growing organ, tissues, skins. Disease modeling. Disease modeling, here we are using animal cells for ascertaining the growth of the disease in human. And how we are going to do it? We take an animal cell, we infect it with suppose today we are battling with the SARS-CoV-2 virus. So, suppose we are going to study about how the SARS-CoV-2 virus is affecting the human. So, we isolate some cells from the human, we infect the cells with the SARS-CoV virus, and we will say, we will ascertain what were the different organs, different cells, how the disease are progressing in the human, to get the knowledge and to develop effective treating method. Vaccine production, and last one is the drug screening. Drug screening, what do you mean by drug screening? Drug screening means we use animal cells to screen various drugs which have been developed into the lab. Earlier drug screening was done using live animals. There are thousands of animals are getting sacrificed by this, because if you have developed drug A, drug B and drug C, out of this only one drug is going to work. So, but you have to test all those 3. So, earlier there were lot of animal getting, lot of mouse, rabbit are getting killed for animal testing. Now, what is happening? We test those drugs onto the animal cells in vitro, and if we suppose we have developed drug A, drug B and drug C, out of drug A, B and C we tested it into the animal cell culture model, and after that we have found that drug B is working and drug A and C is not working, then drug B is gone to the animal model and then to the clinical trial stage. Okay. This is the brief overview of the animal cell culture, where we have discussed about the history, laboratory facilities, cell culture conditions, various system of cell culture, morphology of animal cell in culture, cell counting, characterization, application. These will be discussed in detail into the second lecture. Thank you.